Good morning. It's October 24th, and this is your daily brief in science. Here's everything you need to know. A new antibody has been developed that could change the way aggressive breast cancers are treated. This antibody not only targets tumor cells directly, but also enhances immune responses by activating immune cells both within tumors and in the bloodstream. Researchers have shown in lab and animal studies that this antibody binds immune cells more effectively than current treatments, leading to a stronger immune attack and limiting tumor growth. The team, led by Professor Sophia Karagianis and Dr. Alicia Chenoweth, plans to move toward clinical trials and is working to improve the antibody's lifespan and immune activation capabilities. There is also potential to apply this therapy to other cancers, including ovarian and endometrial cancers. This approach uniquely enhances immune interactions with breast tumor cells, which could be a breakthrough for resistant cancers. The research specifically addresses triple-negative breast cancer, a particularly aggressive form that makes up about 15% of all breast cancers. This subtype is more common among younger women and Black women and is difficult to treat with standard therapies due to the absence of hormone or HER2 receptors. The findings, published in Cancer Research, represent a significant step toward more effective treatments for hard-to-treat breast cancers and potentially other cancer types. Recent research has revealed a new role for hormone-sensitive lipase, or HSL, in fat cells. Traditionally known for its function on lipid droplet surfaces, HSL is now also found in the nucleus of adipocytes, where it helps maintain healthy fat tissue and cellular functions. Elevated nuclear HSL levels have been observed in obese mice, suggesting a link between HSL localization and metabolic diseases. HSL moves between the nucleus and cytoplasm depending on the body's energy state. Low-energy conditions activate nuclear HSL to promote fat breakdown, while nutrient-rich states cause HSL to return to the nucleus through the action of SMAD3. The discovery of this dual role provides new insights into how fat cells regulate energy and could lead to therapies that restore normal adipocyte function rather than simply reducing fat mass. It also helps explain conditions like lipodystrophy, where fat loss occurs despite the absence of obesity due to disrupted fat cell identity and function. Research led by Dominique Langin at the University of Toulouse shows that without HSL, fat cells lose their proper function, affecting mitochondrial activity and shifting from white to beige fat cells that burn energy. This finding challenges previous assumptions that HSL deficiency would cause fat accumulation, instead linking it to fat loss and metabolic dysfunction. Given the global rise in obesity, understanding HSL's nuclear role opens new avenues for targeted treatments for obesity, diabetes, and cardiovascular diseases. Targeting nuclear HSL pathways could provide innovative approaches to address the cellular mechanisms underlying these conditions, offering hope for more effective therapies. A recent study has identified the gene HMGN1 as a key contributor to congenital heart defects in individuals with Down syndrome. Down syndrome is caused by an extra copy of chromosome 21, which leads to overexpression of certain genes. Experiments using human stem cells and mouse models show that increased levels of HMGN1 disrupt normal heart development by reprogramming cardiac cells. This disruption results in malformations that are common in Down syndrome. Scientists suggest that therapies aimed at reducing HMGN1 activity during pregnancy could potentially prevent these heart defects. The research also explores how HMGN1 interacts with other genes like DYRK1, affecting heart development through multigenic and epigenetic mechanisms. The study highlights the importance of combining genomic and computational approaches to understand other chromosome-related disorders, including intellectual disabilities and bone formation issues. Led by Sanjeev S. Ranade, the research demonstrated that overexpression of HMGN1 influences chromatin structure and gene expression, impacting key cardiac regulatory genes such as TBX2 and TBX20. Restoring normal gene dosage by deleting one HMGN1 allele in trisomic cells reversed abnormal gene expression, confirming its role in heart development. While applying therapies like pleiotrophine in humans is still distant, these findings open new avenues for targeted interventions. Overall, this research advances understanding of the genetic and epigenetic factors underlying Down syndrome and may lead to improved health outcomes. Albert Einstein College of Medicine has secured a 5-year, $10.8 million NIH grant to establish the New York Regional Diabetes Research Center. This initiative involves collaboration with Icahn School of Medicine at Mount Sinai and Weill Cornell Medicine, expanding the research scope and team to over 140 scientists and clinicians. The center will serve as a regional hub for diabetes research, education, and training, supported by funding from the NIH's National Institute of Diabetes and Digestive and Kidney Diseases. An administrative core will facilitate pilot studies and help investigators transition to independent research, explore new areas, and develop innovative techniques. Principal investigator Jeffrey Pessin emphasized the center's focus on translating scientific discoveries into clinical care, aiming to improve therapies and understand disease mechanisms. The center will provide mentorship, collaborations, and core services in areas like animal physiology, metabolomics, human islet cells, and organoid technology. Its primary goal is to discover scientific insights and translate them into better treatments for diabetes and related metabolic diseases, including efforts to prevent type 1 diabetes and manage diabetic complications. Research will focus on developing therapies to prevent type 1 diabetes, restore beta cell function, and address diabetic complications. Pessin highlighted that the collaboration with Mount Sinai and Weill Cornell would accelerate translating fundamental discoveries into therapies to improve patient outcomes. The partnership aims to leverage each institution's strengths, especially in beta cell biology and regenerative medicine, to advance diabetes research. Recent analysis of lunar samples from the moon's far side has uncovered fragments of Ci-like chondrites, a rare type of meteorite originating from the outer solar system. These meteorite fragments were identified through chemical and isotopic analysis, particularly oxygen isotope ratios, which act as space forensics to trace their origins. The samples, collected by China's Chang'e 6 mission from the South Pole Aitken Basin, include 7 olivine-bearing clasts that are relics of impact melt droplets. These findings suggest that the moon contains a higher proportion of C1 chondrites than previously believed, indicating they are more common in lunar material than in Earth's meteorite collections. CY chondrites are known for being rich in water and organic compounds, and their presence on the moon implies impacts by these meteorites may have contributed to lunar water sources. The meteorite debris appears to originate from asteroids containing dust older than the solar system, offering insights into the early distribution of volatile compounds. The discovery was made after initial assessment suggested the material was lunar mantle, but chemical and isotopic analysis confirmed its extraterrestrial origin. This research enhances our understanding of material migration within the solar system and raises questions about the moon's volatile inventory and water origins. Since the South Pole Aitken Basin is a key site for studying impact materials, these samples provide valuable clues about the early solar system and planetary evolution. The findings underscore the importance of lunar exploration in revealing the distribution of water and organics, which could influence future lunar resource studies. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.